Hello, everybody, and welcome to this episode of Genomics Lite. My name is Sam.
And I'm Jack. We both work in the Wellcome Connecting Science team at the Wellcome Sanger Institute. Ooh, lovely sound in the background there. Sorry about that. Our roles are to enable everyone in the world to connect with and learn more about genomics. If you haven't joined us before, then Genomics Lite is a series of short sessions that share the science that happens on our campus, which is all about DNA codes. This is our second video of our spring series, exploring some key questions about genomics that we get from people like you.
And the series is streaming live on YouTube right now. So welcome if you're joining us on the live stream. And all of our Genomics Lite videos are available afterwards on our YouTube channel. So do go and check those out after this video. Don't go yet. But in this session, we're going to explore antimicrobial resistance. Now, anyone who joined us back in January now, that seems like a lifetime ago, will remember that we covered a little bit of a slightly more local example of antimicrobial resistance with one of the scientists on this site, Honor, working by studying antimicrobial resistance in the River Cam here in Cambridge, which is where we are based today. In this session, what we're going to be doing is looking at antimicrobial resistance, but we're going to be taking a far more global view and speak to two different scientists about how resistance occurs in different settings and how genomics can provide some tools to enable us to monitor, understand, and also treat the disease as things change and adapt. So, Jack, what do we need to know before we introduce our two wonderful guests today?
Well, first, let's get a better sense of what this thing is that we're going to be talking about today. Antimicrobial resistance. It's a big word that's made up of three different parts. So firstly, microbial. We're talking about microbes. We're talking about microscopic living organisms. And these organisms come in lots and lots of different kinds. Some of them are positive. Some of them, like those that live in our gut, help us digest our food, have good impacts on human health. But there are others that are what we would call pathogens that cause disease and illness. And many of the viewers will be familiar with lots of these. These are the bacteria, fungi, and viruses like tuberculosis or athlete's foot or influenza. And today we're focusing on those bad ones. So that's microbial. Antimicrobial. So antimicrobial are all the different things that we do to fight these bad, negative microbes. So that might be medicine that we're taken into our bodies, like penicillin, or it might be the kind of cleaning fluids that we use on our kitchen surfaces, or when we have a cut on our skin that disinfects or that gets rid of those microorganisms. So that's antimicrobial substances. And these work in different ways. So finally, antimicrobial resistance is when those microorganisms change or evolve such that those antimicrobial substances, those things that we've found or designed to kill these bad microbes, they've changed so that those don't work anymore. They've developed resistance. And that can also occur in a few different ways, and they're going to be the things that our guests take us through. So we're talking about microbes changing so that drugs or cleaning products don't work on them, and how studying DNA can help us to detect or predict those changes and adapt our strategies to keep people safe. You might have heard this word superbug before, and that means a microbe that's changed so that lots of different drugs or treatments that we might use don't work on it anymore, and that means it's really difficult to control. These things are really big problems for human health all over the world.
Okay, well, as you said, it's a big topic. We've only got 45 minutes, of which 5 minutes has already gone. So let's take this opportunity to introduce our first guest. So it gives me a great pleasure to introduce Nisha, who is a postdoctoral fellow in the Thompson Group in the Parasites and Microbes team here at the Wellcome Sanger Institute. Now, Nisha's done a lot of work out in India sampling and monitoring cholera outbreaks. So cholera is going to be something that we're going to start here with you, Nisha. So it's lovely to have you, Nisha. Perhaps could you start by telling us and our audience watching today a little bit about what cholera is and how we treat it, what a large problem it is, and then we'll take a little bit deeper dive into where antimicrobial resistance comes in. But yeah, introduce cholera to us.
Thank you very much, Sam, for your kind introduction, and a very good afternoon to everyone. So cholera, we call it like a disease of poverty. So it affects the people who are living in a place in a community where there is no proper water supply and no sanitation. So how they get that infection? If they consume food or water contaminated with a bacteria called Vibrio cholerae, which causes cholera, so they get the infection. And when they have this infection, so there is a like a release of fluid in like a fluid, loss of fluid. They have multiple episodes of diarrhea and vomiting, and if it does not get treated on time, so they can die as well. So yeah, it's very threatening if you don't treat it on time. And for treatment, it's very easy actually. So what is there is no antibiotic recommended for treatment if they are having any mild and moderate infection. There is just ORS, which we call oral rehydration salt. If they take it, then it can replace the fluid which you have already lost, and salt, and then you can be cured. But in case if you are having a severe infection and if a person is immunocompromised, then in that case they have to take antibiotics, otherwise not. Yeah.
And our session today is looking at antimicrobial resistance. Give us, in the context of cholera, something that you have, been out in the field sampling and stuff and monitoring. Give us a sense of what does antimicrobial resistance mean for something like cholera?
As you have explained, so I will explain in a very simpler language actually. So for example, if you are having headache and you are taking any medicine for that, then it works. Then you having headache again, you are taking medicine again, it works. But over the time, it stop working on headache, then your body might not respond for that particular medicine which you are using. It's same in case of bacteria as well, that the drug you are using for treating that infection might not work at certain period of time because bacteria develop resistance for that antibiotic. So in case of cholera, I will not say that it is a big major problem till now, because there are multiple antibiotics which you can use to treat cholera. But there are examples when Vibrio cholerae has developed resistance against some of the antibiotics. So there is a big outbreak in Yemen. There is a study published from our group as well about that cholera outbreak. And what happened, so it started around October 2016, that outbreak, and they were using macrolide, azithromycin, and erythromycin for treating pregnant women and the children's. But what they noticed that over the period of time, like after around 2019, those antibiotics, they stopped working against the disease. And then they found that there must be something with the Vibrio cholerae, that they may have acquired the resistance for that antibiotic, and they can't use the same antibiotic anymore for treating that infection in that area.
And it might be a concept that would be great to sort of get from you in terms of how does this bacteria start to develop this resistance? What is actually going on there where you say an antibiotic, a drug that is administered, works and works and works, and then it doesn't. So what is happening there with the bacteria mean that it becomes resistant?
So there are, in general, there are like multiple things which can kind of promote bacteria to develop resistance. First of all, there is like overuse of antibiotic, whether you you need it or don't need it, but you go just take over the counter available antibiotics and you take it. For example, somebody is having viral infection and they just go to the counter and take antibiotic, and which is not recommended, because you are because viral infection you are not supposed to take antibiotic. And so for bacteria there are certain mechanism how they can develop antimicrobial resistance. And so if you when we treat bacteria with when we take antibiotic, so bacteria has a tendency that some of the bacteria they will be killed, but some of the bacteria they are really tough and they might survive because if you take that antibiotic, and these bacteria might be responsible for spreading this antimicrobial resistance to the other bacteria when they come in contact with those bacteria as well. So if you look at the mechanism of the antimicrobial resistance, how they develop it, there are multiple ways. So bacteria has a particular thing, we call it like a structure like efflux pump. So if you are providing antibiotic for treating an infection, bacteria might throw out that antibiotic out of his cell. So we can't use that antibiotic. And there is another way that they might the antibiotic might enter to the cell, but they have mechanism, they produce some enzyme and that can break the antibiotic as well. And they can also modify antibiotic. For example, if they can modify themselves so that the antibiotic is not responsible for those bacteria. For example, if you change a lock, and then the key is not going to work if they are modifying themselves as well. And most important is that gene. So they might develop some features that those genes, and if one of the bacteria having that antimicrobial resistance mechanism, it can transfer it to another bacteria which it is coming in contact with.
And we started to in that session that we mentioned in January slightly unpick this concept of horizontal gene transfer.
Yeah.
And that's what you mean when you say sort of these genes passing between different bacteria.
Yeah, that's true. Yeah.
And just for our audience as well, if there was is that through the same types of bacteria, or can these genes be spread between other bacteria as well?
Yeah, so if I'm talking about Vibrio cholerae, so like other organism, for example, E. coli, which is like which cause disease in the similar way as Vibrio cholerae, because through the ingestion of contaminated food and water. So if the water is contaminated, they live in the same environment. So it is not necessary that Vibrio cholerae will transfer antimicrobial resistance only to Vibrio cholerae. There might be chances that it may transfer antimicrobial resistance with E. coli or maybe some other bacteria, Klebsiella pneumoniae, which is present in the same environment. So it's just not restricted to one specific bacteria. It can spread across different genus of bacteria, actually.
So I think what we've, as Jack mentioned in this episode, what we're trying to get across is and try and unpick a little bit is that sort of global perspective of antimicrobial resistance. And I think what you're starting to open there is just how, you know, monitoring in say one strain of or one type of bacteria is really important, because it has knock-on effects potentially in if they share the same environment, then that can have an impact further down.
Yeah.
Now, obviously, we study DNA on the site that we're at. Just give us a sense at how you use genomic science and DNA-based science to learn more about, say, cholera and how that's spreading. But yeah, what is DNA helping you to do there?
Yeah, so we call it like term genomics, which is a key actually, which is really very important. So genomics is like a study of genome, which is nothing like DNA. Genome is, we call it DNA. So we can do a detailed study about DNA, like what are the mutations, like where there is a changes occurring in the DNA of the bacteria, which is being responsible for causing this antimicrobial resistance. And with the help of genomics, so we can trace it, that where it has emerged particular reason. For example, it has emerged in particular hospital, whether it has emerged in particular city, particular country or continent. So that's how we can track, and then we can take, like on the basis of these genomic information, we can take preventive measures, so that, because prevention is better than cure. So if you prevent it in that particular region, so it might not spread to other region as well. So what we do, for example, if somebody is having multi-drug resistant bacteria, like having bug which is like resistant for multiple antibiotics. So in generally, if there is a like a in a hospital, what they do, they keep that person separate from the other patients, so that it don't spread among other people as well. So that's why genomics really is really very important to check where these changes are occurring, what mutations are occurring, whether this is just only for one antibiotic or this is like for multiple antibiotic. As I said about Yemen, so that bacteria acquired a particular plasmid which is not causing responsible for the antibiotic they was using, erythromycin and azithromycin macrolide, and that bacteria become multi-drug resistant and causing like a resistant for other antibiotics as well, not just macrolide. So
you used a word there, Nisha, that some of our viewers won't know. Could you just describe what plasmid means for people?
So it's like it's DNA only, but it's like it can circular DNA, which can be transmitted among different bacteria. It carries mostly antimicrobial resistance genes, and which can be acquired from multiple bacteria, because plasmid, same kind of plasmid can be found from between different bacteria. Maybe it can be found in E. coli, it can be found in Klebsiella, and it carries antimicrobial resistant genes from E. coli and Klebsiella as well. And then it can be transferred from one bacteria to another bacteria through horizontal gene transfer, as Sam has mentioned earlier.
So these are circular bits of DNA floating around within a bacteria that can go across into another bacteria in a separate cell over here. Is that right?
Yeah, that's true.
Fantastic. I had we've had a question here from one of our audience members ahead of this session, which was about the process by which these changes in the DNA that you've mentioned, these mutations occur that create drug resistance. And the question was, they said, I would like to know if antibiotic resistance is a purely random process. Just to flag for viewers, they've used antibiotic resistance here. Antibiotics are antimicrobial medicines. Antibiotics means ones that we take into our body, so those kind of medicines that we swallow or inject. So their question again was, is antibiotic resistance a purely random process, and how does it develop? Could you talk us through that from a DNA perspective?
So definitely we are trying to understand that, because different bacteria lives in a different environment where it is particularly occurring, whether it is in environment, it is just maybe in human beings, where in animals. What is the major driver for causing antimicrobial resistance? It's like a selection pressure. So bacteria is living in an environment where it is living in a harsh environment, and until and unless it modifies itself, mutate DNA and other things, so that it can survive those harsh conditions. So that's how they develop antimicrobial resistance to cope up in this harsh environment, actually. Yeah.
So from the DNA perspective, there'll be changes that are happening all the time. We'll swap a letter here or a letter here as the bacteria evolves and reproduces. Only some of those would create any kind of resistance to antibiotics or antimicrobial chemicals. But because of the pressure those chemicals put on the bacteria, they're going to kill them if they can. any of those changes that do lead to that resistance are going to multiply and are going to really succeed. Is that correct, Nisha?
Yeah, that is possible. It's just not, I'm not saying it's just the plasmid which is being responsible for causing or transferring resistance among different bacteria. There may be some insertion elements like a small part of DNA. So if we talk about horizontal gene transfer, there are 3 different mechanisms for them. So there, I think it's going to be very deep, but it's like transduction, translation, and conjugation. So conjugation is just when 2 bacteria come in contact with a structure which attach them, and then they can transfer the DNA among them. And then there is a transduction. When there is a virus involved, we call it like a bacteriophages, which can kill bacteria. So they acquire some part of the bacterial DNA, and then they infect another bacteria. So this part from this bacteria can be transmitted to the transferred to the another bacteria. And then we have transformation, where there is like a naked DNA present in the environment, and bacteria can acquire that. So in these ways, bacteria can acquire AMR genes, which is present in these small parts of DNA, which is present in the environment through bacteriophages or through acquiring naked DNA or conjugation as well.
And just to kind of pin it to this question, in theory, those processes that you've described there, the mechanisms are random, but because of these pressures, it doesn't appear random, really, does it? They're kind of the ones that will succeed and proliferate are the ones with these resistance genes. Is that correct?
Yeah.
Great. Thank you, Nisha. I think this is a great point for me to introduce our second guest, to bring in another perspective here. So thank you, Nisha. Please stay with us. But Adrian, I'd love to introduce now. Adrian's a staff scientist working in the Thompson Group as well, in the Parasites and Microbes team here at the Wellcome Sanger Institute. Adrian and the team that Adrian works in have been looking at exploring the evolution of bacteria through time. So that involves historical bacterial samples. And Adrian, I wonder whether that's the best place for us to begin. So welcome. It's wonderful to have you here with us. What do we mean by historical bacterial samples? What is the material that you're working with?
Right. Well, first of all, thank you for having me. I'm really happy to join the conversation. Yeah, with historical archives, historical isolates, what we mean is these are bacteria that were collected in the past. In this particular collection we're working with, we're speaking about anywhere between 1917, 1954. So these are bacteria that, just like the one that we face today, cause infection, but the microbiologists and scientists back then were interested in these ones, so they collected them, they saved them, and they froze them down. And so they've been frozen ever since up until now. So that's what we mean with historical bacteria, something that was collected and then frozen down in the past.
And 1917 is a long time ago. They would have had very different scientific understanding then, different kind of techniques. What is it that you can do now with these samples that the scientists weren't able to do in those decades past?
Yeah, I mean, that's a very important point of why it's important to collect these ones and then preserve them. As you say, science moves forward very quickly, and usually that's prompted by technology. And very in the context of this conversation is genomics one of those ones. So one of the things that we couldn't do back then is decoding all the DNA that was in these bacteria, a technology that emerged eventually and that now everybody uses, and in campus that's what underpins our research. So one of the things that we can do with those ones that we couldn't do before is determining this genetic code in those bacteria that existed in the past.
And we've heard a brilliant picture from Nisha about how some of these changes occur within the cholera samples and some of the other kind of microbes that Nisha mentioned earlier. What are the kind of mechanisms that you're seeing that are causing changes in the samples that you're working with? And perhaps just name a couple of the kind of different pathogens that you're working with in these historical samples.
Yes, so I think if that's okay, I would more or less introduce one of the main reasons why we started working with this collection in the first place, that links, I think, everything together. I think there's this misconception around the origin of antibiotics and antimicrobials in general. Where do they come from? These molecules existed way before humans used them as therapeutics, because bacteria always coexist with other microorganisms, and they are always fighting, they are competing with each other, right? So eventually in the 1940s, humans discovered antibiotics and started industrializing their production. And that was in the context of the Second World War. And what happened at that point is that the world changed, and the world has never been the same at that point. But what is really very interesting about the world and this collection in particular is that the world changed in two contrasting ways. One is visible and one is invisible. The visible one was modern medicine really started at that point, because antibiotics really underpin modern medicine. So in terms of the human side of things, antibiotics save millions of lives, and they continue to do so. But then you have the invisible way, right? So the invisible way is what happened with all of those bacteria that were there, minding their own businesses, and then we industrialize and then release these massive amounts of antibiotics. So that is with this collection really comes into place, and try to link to the question that you asked. So at this point, we can only imagine what happened with that bacteria. And if you really use your imagination, you can think of this as it was probably the blitz for bacteria, right? So all of a sudden, living again, minding their own business, they are constantly, intensively bombarded by these antibiotics, and that bombardment causes damage. And then it damages the cells and damages infrastructure and kills most bacteria unexpectedly. A few survivors probably are the ones that adapted the best, and then they can survive in now this very harsh environment, right? So by looking at this collection, we can understand what happened in the past, how this population of bacteria looked like before they were exposed to all of these antibiotics that we dump. And specifically related to the question that you say, these are incredible resources because they are history genetic books that tell us how the DNA was in the past. And so now we can compare it to bacteria that we see today. And now, finally, answering directly your question, we know what mutations make bacteria resistant in modern pathogens. But one question you can ask is, were those mutations already present in those bacteria before we use antibiotics as therapeutics? How they change all of this time. And what we found thus far, because the research is still ongoing, is these bacteria already have some of the resistant genes that we see today. And yet bacteria were sensitive to antibiotics back then. So this collection is sensitive to almost every or all antibiotics that we use today. So some of the mutations that we've seen in these bacteria are mostly enterics. So we find Escherichia, Klebsiella, Proteus, Salmonella. You've heard of all of these bacteria in the past. So they are the same pathogens, and they sometimes have the same genes. They just weren't resistant yet. So now we're trying to define how things change so that now all of these pathogens that we face today are resistant.
That was a beautiful description. I've never heard it described in that way before. I want to linger there a moment of this world outside of human beings. We're so self-centered, really, aren't we, in the stories that we tell, that and I imagine many of our viewers too, really pictured this as a conflict between us versus these pathogenic microbes, us versus the diseases that make us sick. What you've described there is actually that's not it at all. These things have been in conflict with each other for millions and millions of years. And we're a very late part of that story. And it just so happens that since 1940s, we've been producing these things that really impact on those populations. So my question I wanted to return back on was one that you posed partway through that description, Adrian, which was, were these things, these elements that have allowed these microbes to become resistant now already existing in those populations, or were they modern mutations that have happened since the introduction of antibiotics or antimicrobial usage. What's the answer to that? Were these things already existing? Do we know the answer to that yet?
Yeah, we do. I think in essence is a combination of both. I think the point that you make, that this war between microorganisms has been going for way longer than us fighting bacteria as pathogens, meant that many of the resistant genes that we find today were already there in those populations, because bacteria already have to survive these encounters with their enemies and just survive those. So some of those genes were already there. But then what has happened in the last few years is that the selective pressure, which means how much of that chemical you find in nature, now has increased so much that from this starting point, the genes that you already have, bacteria have perfected them. So now they accumulate mutation that makes them much, much, much better, and that now fit for purpose in modern days. So it's been a combination of both. Bacteria acquire these things that are already around the microbial world. And I think you both already mentioned that bacteria can share genes, so they can already come ready to be used, but then they need to be perfected over time. And what we face today is very much the optimal function of those genes. So I think it's a mix of both, what we've been discovering across different pathogens.
Amazing. Thank you, Adrian. Now we've sort of introduced I think you've both really given us a really nice picture of, back also from how bacteria have evolved, but also what are current sort of outbreaks, as Nisha, you mentioned, in things in Yemen currently, and the papers that you've written about recent sort of outbreaks of bacterial pathogens and stuff like that. Give us a sense of you two, both of you as a group, working your team. What does the information that you are starting to glean allow us as a society to be able to start to do? And for example, in the clinical setting potentially, or you as scientists or even policymakers, what does the information that you are gathering as a team allow us to do? Nisha, I wonder if I can go to you first on that.
Yeah, so from genomics aspect, it's really very important. For example, for us who are working in lab, who are just working on computers, or who are working doing some wet lab work, it's really very important, because now we are seeing on the basis of that where this antimicrobial resistance is occurring. It's just in one bacteria or maybe multiple bacteria. So what can be the mechanism of antimicrobial resistance as well? So for that aspect, it's really very important as being a scientist. If we talk about clinicians, so because now we know that, this bacteria is resistant for this particular antibiotic, if I'll use that antibiotic, so it's not going to be effective for patients. So they can change that antibiotic, and they can use another antibiotic which is bacteria is sensitive for that antibiotic for treating those patients. And if we talk about the policymakers, so genomics keep us like a one step ahead of maybe I can say bacteria actually, because of that, because we will be knowing that, these are the mechanisms, this is the particular bacteria having this kind of resistance for this antibiotics. So on the basis of that, policymakers, they can introduce public health measures to prevent those infection, and then prevent the spread of those infection in a larger population as well. So it's important for on all the levels actually.
Just a question that's kind of popped into my head there, and maybe might be of interest to our viewers, which is just, you said you as a team, you might discover that through your sampling and understanding the genes that a certain bacteria has, say, developed a resistance to a drug. What is the sort of speed from you learning that type of information for it to then sort of see that impact in, say, a clinical setting where a clinician knows that antibiotic is no longer usable, shall we say, for that bacteria? How quick is that sort of implementation?
I think now it is now genomics is just not limited to the big institute, for example, Sanger, because people now can use, for example, other techniques like MinION, where they can sequence the bacteria if the bacteria is not responding to the antibiotics they are using. Though they do they perform like a conventional test like antimicrobial susceptibility, where they can know that which antibiotic is working or not. But in some cases, MDR or extensively drug-resistant bacteria, you might not be able to find antibiotic which is being used for treating that infection. So that's why genomics plays a very important role. Then you can screen the DNA of that particular bug and see that what antibiotic resistance genes it is carrying, and what are the antibiotics we can't use for treating this infection anymore.
Mm. And Adrian, I want you to give me a sense of kind of, and I think you alluded to in the sort of historical samples as well that you mentioned, but give me a sense of, yeah, just give us a sense of that global context of how sort of, I don't want to be like, I don't know how to phrase this question without being ensuing worry amongst our audience, but like, just tell us, what is the global picture of antimicrobial resistance, and how concerned should we be, and what should maybe we think about that might change our perspective of antimicrobial resistance?
Yeah, thanks for the question. And this is a very important one. I mean, unfortunately, there is not an easy way to put this, because the picture really looks bad. And I think that the main issue is that we don't seem to see it close. I mean, there's a reason why this AMR global crisis is also called the silent pandemic. And it's basically because you don't seem to see it closely to you. But if we just look at the numbers, just to put an example, I think the WHO has estimated that there were over 5 million deaths related to antimicrobial resistance, which, you put it in different contexts, that number is basically almost the whole population of Norway. So that would be the amount of people that you'll be losing to antimicrobial resistant infections every year. So the numbers are already just bad when you look at them. The big problem is that this spreads very rapidly. And so that doesn't mean that that will continue happening in following years. That will increase, it's gonna get worse. And then everybody is affected. And I think that is the main issue. At this point, we know that bacteria are increasingly becoming resistant to more and more of the antibiotics that we have. So the picture really looks bad if we don't do anything about it, and it affects us all. So I think that is the main issue at this particular point in time, that we don't seem to be worried enough. And this is not a cause for people to panic, but it's a call for awareness and action, that we really need to do something before we can get to that point that there's no return. So I think it's a very important question, and it needs to be discussed.
And I guess, as you mentioned, there is often where people sort of connect with antimicrobial resistance is, say, if they are unwell and they are put on, say, a treatment of antibiotics or something like that. How do you, this might be a bit of a tough question to ask you, but how do you see maybe us shifting that mindset in that sense of, it's not just at that point, it's sort of all around in the world or in the environment?
Yeah, I mean, one of the main outcomes I've seen at a high level from the different research areas, and genomics has been key to this because of what Nisha said about cholera and environment, is that we have now developed this One Health approach, where we know basically One Health means we acknowledge that animals' health, humans' health, and environment health are interconnected. So one affects the other. So the first step is to understand that this is not just about humans, it's about everything that is going on around us. And it's these two ways effect, that how do we affect the environment, and therefore how the environment affect us. So then the first step now going forward is that we have recognized that problem, and then we realize that even if we get to put all the money we had funding AMR research in humans exclusively, that will not stop the problem. We need to look at how we pollute environments, that would relate to what Ona said in that podcast, and also animals' health. So I think now the point moving forward is we have recognized it's a complex problem that is interconnected, and we need to all push together in the same direction if we really want this to change.
I want just a short comment there on animals that you've mentioned. We've been speaking about human diseases so far, and I think most viewers will think of medicine and drugs being something that we mainly for humans, but of course in our agricultural sector it's very important too. Perhaps Adrian, I'll keep this one with you. Just give us a sense of AMR within a non-human context, so resistance in animals that surround us.
Yes, antibiotics are commonly used in agriculture and farming industry as well, but also aquaculture. So basically, we try to stop these animals that we eat from being infected. There was a big problem in the past where we assumed that antibiotics would also promote growth, and then we started overusing them in farming. Antibiotics are heavily used for veterinary medicine as well. So basically, antibiotics are a part of almost every single thing that we do as humans. We just don't see it. And everything ends up eventually in the environment, whether that's soil or water, whether that's wastewater or rivers. So it just circulates all across. But those are the main usage. And obviously, in research and development, pharma will also develop new chemicals and stuff that eventually make their way into the environment. So many different ways that we use them that impact different areas.
Nisha, I wanted to come to you and to bring us back to cholera, where we began, for I think a sense of pace of change here. We heard from Adrian, you know, it's been since the 1940s that we've been using these antimicrobial or antibiotic medicines. And that isn't a very long time for evolution when we think of it in the kind of large creatures like humans or birds or the things that Darwin described. Why is that different for cholera? How can these things evolve so quickly, you know, in the space of 80 years, as we're talking about there?
If you look at the bacteria, like it just takes 20 to 30 minutes to like multiply. So one cell will like become 2 cells, then 4 cells, then 8 cells, and so on. And then because the rate that how fast it is like multiplying, so chances of spreading antimicrobial resistance across these bacteria and acquiring, and how in different environment bacteria is being found as well, how it come in contact with the other bacteria as well, how often. So these all factors are responsible for like evolution in bacteria. So it's quite quick actually comparison to human beings and other big creature. Yeah, that's what.
And those mechanisms that you described, the horizontal gene transfer, the plasmids that we got into early on, that isn't something that I hear a huge amount in terms of human evolutionary mechanisms. Could you linger there a little as well? What do we see in there that's different in these microbes, and how come that impacts on their evolution differently?
Again, I'll say that where it is living actually. So as Eden mentioned, that the selection pressure plays a really very important role. So which may kind of force bacteria to have these kind of mechanism more often to acquire some of the external genetic material which can help bacteria to survive in those selective pressure environment where there is a stress. So definitely bacteria have that advantage actually to acquire and then gain and then kind of evolving over time. Yeah.
We've got a sense of challenge. We've got the global challenge, Adrian, that you've described, and this pace of change that can happen within these populations. What about our pace of change now? Just to finish, as I know we're drawing to the end of our time together, Adrian, how is the science and the understanding that we have changing in this competition that we have against antimicrobial resistance?
Yeah, I mean, in the simplest way, AMR is just life finding its way. And it's really just this push for survival from the microorganism, which is the very instinct that we all have as living things. So we have really moved very quickly now with the technologies we have. And if something we learn from COVID is how quickly we can go if there is will and if we are all focused and we have all of these amazing tools, from understanding all the different strategies bacteria use to survive these antibiotics to then find new ways to stop them from infecting us. So now most of the research really is divided into one is how quickly we can identify these microorganisms, and the other side is how quickly we can identify other targets and develop new drugs and develop vaccines, because now we have full access to the genetic code. So it's moving very fast and it's going in the right direction.
That feels like a positive note to hang on to, though still with that warning, of course, in our ears. We should not be complacent. We're making progress, but arguably not fast enough. And we need to speed up research. I hope that means that those viewing will be looking at careers like Nisha's and Adrian's and will be coming to join us working at the Sanger Institute to tackle these big global challenges in the future. I'm afraid that's all we've got time for today. I know there's lots more to discuss with antimicrobial resistance, and I'm sure it's something that we'll return to again on a future episode. So thank you very much, Nisha and Adrian, for a brilliant discussion with us today.
And I'd like to thank everybody who has watched us today. It's just worth saying before we close the session that we are back next month, and hopefully what these sessions start to do is to build trust in science, and that's what we're going to be looking at next month. We're going to be looking at trust in science. Why should we trust science, and what is the importance of trusting science? So we're going to be unpicking all of that next month. So do sign up. That is happening after this webinar closes. You can sign up for that. But thank you everybody for watching, and thank you finally, Nisha and Adrian.